Hey guys, welcome to your channel Jeevashastra Abhyasa. I am your educator Niranjan Hegde. So in this video, we are going to discuss about messenger RNA, which is also abbreviated as mRNA. In the last video, I have asked you a question, which is the official currency of United Kingdom, and the answer is pound sterling. It is also known as British pound. So for this video, a new question is, which is the official currency of Bolivia? I repeat it once again, which is the official currency of Bolivia? So you have to comment below to give the answer. So then what is the wait for that? So let's begin. Before learning the structure of mRNA, we have to know what is RNA. RNA. It is also abbreviated as ribonucleic acid. Ribonucleic acid. So then you may ask me, what is the main difference between RNA and DNA? So as you can see over here, so there are 4 carbon atom is there, 1, 2, 3 and 4. So which is attached with the hydrogen molecules. Now it's been 5 carbon rings are there. This is a sugar molecule which is attached with nitrogen base in the first position, and in the 5th position, so phosphate group is attached, and also so while forming the polynucleotide chain, so in for the 3rd position also the phosphate group may be attached. So next moving on to the next, that is so what is mRNA and how it has been different from RNA? So it is not a different from RNA, it is one of the type of RNA. RNA has, the ribonucleic acid has 3 types, the 1, 2 and 3. First one is rRNA, second one is mRNA, and the third one is tRNA. So then what are these things? So this is a ribosomal RNA, and this is a messenger RNA, which we are going to study today, and this is transfer RNA, which we are going to study in the next video. So today, as we are going to study the mRNA, so we will move to the topic mRNA. So which is a term abbreviated as a messenger RNA. So the name itself suggests it carries some messages from DNA towards the cytoplasm, and mainly the synthesis of protein, the site of synthesis of protein. It carries some messages for the generation of proteins. Now the mRNA was mainly found by Walkin, the scientist named Walkin. So in Escherichia coli bacteria, and which was named as mRNA by Jacob and Monod in 1961. I hope you got the point. And so this is mainly a single stranded structure. It is a single stranded structure with around 1000 to 2000 nucleotides. It has 1000 to 2000 nucleotides approximately. Okay, and it has also been divided into 2 types mainly. So the 2 types are, one is monocistronic, which can only be found in eukaryotes. So it is only found in monocistronic is type of mRNA is found in eukaryotes. So I will show you what it is there when I am drawing the diagram. So next thing, so here it is. So it can code only for one protein, codes only for one protein, codes for one protein, and also a long lived, and the second one, coming to the second one, that is polycistronic. So this is found only in prokaryotes. So which codes for several proteins at a time. That means the bacteria can produce a number of proteins in a single synthesis. Okay. Then coming to it is a short-lived one. As bacteria itself, it is a short-lived, and also it is mainly, I am telling you here, that is it has a very big number of nucleotide, that is around 3000 to 8000 nucleotides you can found in the polycistronic type of mRNA. So now we will see the diagram of both of them, and we are going to study about the monocistronic type of mRNA. Coming to the structure of monocistronic mRNA, it looks like a straight scale which has been divided into 6 different positions, or the stages we can say. So let's study first. We need to study the monocistronic mRNA. Then I'll tell you how the polycistronic mRNA looks like. It is composed, as I told you, it is composed of 6 different positions from cap to the poly A tail. Okay, so then let's learn what is the function and what are those 6 different positions. Coming to the first position, that is cap. It is known as cap, which acts as a protective layer of mRNA. So which is formed by 7-methyl guanosine. Remember that it is made up of 7-methyl guanosine, 7-methyl guanosine nucleotide, which is attached to the nitrogen base of another, nitrogen base of the mRNA strand. Okay, so with nitrogen base. So it can also be termed, this can also be termed as M7G 5-PPP 5-N. Okay, so it is also can be termed as like that, 7-methyl guanosine with nucleotide which is attached to the nitrogen base with phosphate end. Okay, 5-. So as mRNA you can see it starts from 5- to 3 and ends with a 3- end. So coming to the next position, this is the leader sequence. It is known as what? Leader sequence, which has 100 to 100. It has 100 to from 10 to 100 nucleotide. I made it ulta. So 10 to 100 nucleotides. It has 10 to 100 nucleotides, which is also known as a non-coding region. It is also known as non-coding region, that is termed as NC1, the first non-coding region it is, so which is known as leader sequence. Coming to the next position, that is the third position, that is initiator codon. So this is the codon which is responsible for the synthesis of protein synthesis, that is AUG, always it will be AUG or rarely GUG, AUG or rarely GUG. Moving to the next position, that is a coding region. So here the main message which is prescribed or which is transferred from DNA is present in this particular region, which has around 1500 nucleotides. 1500 nucleotides. In 1500 nucleotides, there will be message for the synthesis of a protein molecule. So when the protein is generated, so it cannot continue for a long time. So that's why there is a stop for this particular protein synthesis there, and that on the mRNA itself, and that is terminator codons. There are 3 types of terminator codons are there, as you all know that is UAA, which comes frequently. UAA comes very frequently, and next is UGA, the second one, which is a little bit, it comes a little bit, and the last one is UAG. These are the three different terminator codons which ends the synthesis of protein generation. Then moving to the next part, that is the NC2 region, that is NC2, that means non-coding region 2. So that means one is before the initiator codon and another one is after the coding, after the termination codon. There is non-coding region is that, which has 50 to 150 nucleotides, which doesn't code for any of the amino acids. So coming to the last one, that is poly-A tail. So what is poly-A tail? So it is a several number of adenylate. So adenylate nucleotides will be there, several number. So which forms a tail-like structure. And until now, there is no confirmation of function has been given to us. And this particular poly-A tail is not present in the prokaryotic mRNA, which is also known as polycistronic. Now coming to the function here. So in the region coding region, so this is the main region, as you all know, that protein synthesis happens. The message which is present over there will be transcribed into a protein. So that protein will be it is a polysaccharide content which is formed by the several number of amino acids together. So all the nucleotides are in combinations of three set of codes. They are known as codons, that is AUG, AAA, whatever the codon may be. For each protein there is a particular set of codons will be present on the coding region. So this is the structure of monocistronic mRNA, which is important for in the way of examination purpose also. Now coming to the polycistronic mRNA strand, that is, it looks like a single stranded DNA which has all the structures, only it will not contain any poly-A tail. The sixth position it doesn't contain. It also starts with a 5' end and ends with a 3' end, which has all the six regions, leaving the seventh one, that is poly-A tail. And this is of the mRNA structure. If you like this video, please share, like and subscribe to the channel. So that is very important, because you can get more number of videos, which will push me to create more videos. And also press the bell icon to get notification whenever I put the video into the YouTube platform. Until next time, let's hit the road of success together. Jai Hind.